Welcome, everybody. And although it's sad we're not physically together, I've got to say, this is pretty great. And it's wonderful to welcome all of you. And I'm not sure whether they're supposed to, certain Hermann is here from Germany. There may be someone from France. There's someone from California coming, Florida, Connecticut, New Jersey, New York, and Maine. So there's something quite amazing about it. So anyway, let me be formal, and on behalf of Anna Maynard and Rob Whitman and myself, which is the President's Cabinet of MDI-BL, we're really happy to welcome you to the first virtual conversation with Hermann Haller about his work, Cloaked in Sugar. Today, you'll hear a story about how years of dedicated scientific research in one area has led to an unexpected and relevant discovery in another. And this is just one example of the exciting and brilliant research that's been being done by the scientists at MDI-BL. There's the scientific research there, and the educational programs are just extraordinary. And yet, I've lived in Mount Desert summers all my life and literally recently discovered Mount Desert Island Biological Laboratory. So it's really one of the best kept secrets on the island. And so here's this. If you like what you're about to hear as much as I think you will, then I hope you'll tell others about it, and not let it remain a secret, because it's too good to be a secret. So anyway, let me introduce Jerry Bauer, who's the Director of Development and Public Relations, who will moderate tonight's discussion.
Jane, thank you so much, and welcome everyone. It's so much fun to see all your faces. It's been a little quiet around here, as you can imagine, so it's just great to see you, and I'm so pleased that you've joined us this afternoon, and I think we're going to have a great conversation, very timely and interesting conversation with Dr. Haller. And Jane, my thanks to you and Anna and Rob for your willingness to share your enthusiasm for the work the lab is doing and for bringing all of us together. It's just a wonderful, wonderful opportunity. Thank you both, thank all of you so much. And thanks to everyone again for being with us today. It's a gorgeous afternoon here on the coast of Maine. And as Jane said, it's a shame that we can't all have you here on campus, right on the shore of Frenchman Bay. But we are excited and grateful for the opportunity to be with you via technology and share this virtual experience with you. It's clear that we're living in turbulent times, and COVID-19 has certainly profoundly impacted our daily life, and it's likely to do so for the foreseeable future. And while there's no lack of information out there about this virus, sometimes it is difficult to be able to really determine which sources are reliable and trustworthy. And so that's part of why we're bringing everyone together today to have an opportunity to speak with Dr. Haller, who has been mentioned, is a physician scientist. He's on the front lines of seeing patients who are suffering from complications of COVID-19, but he's also a research scientist. And so he brings a unique perspective to this problem, and I think is a great source of information. Certainly he has been for all of us here at MDI-BL as we learn to navigate this complex world that we're all living in. So we're excited to be able to share that with you today. Let's see, I do want to make a special welcome to those of you who are joining us for the first time. I know that I see lots of familiar faces out there, but there are many of you who maybe haven't yet had an opportunity to visit the MDI Biolab. And so while we're going to hold you to an in-person visit as soon as we can, in the meantime, we're excited to give you this virtual glimpse at some of the work that we're doing. The lab is an interesting and complex place. And today we're going to focus on, one aspect of our research, but there is a lot to share about this organization. And as Jane said, it is kind of one of those hidden jewels here on MDI. I've been at the institution now for 21 years, and if I had $1 for every time that someone said, "Oh, are you the Jackson Lab?" I wouldn't need to be a fundraiser anymore. So it's a great place, but it is kind of unknown. And so that's part of why we are holding these kind of sessions, to really just increase awareness about the exciting work that's happening here at the lab. And so by way of introduction, I want to just tell you a little bit briefly about who we are and what we do. The lab is actually 122 years old this year. It was established in 1898 in South Harpswell, and it was originally a summer school for biology students from Tufts College. And it was at the behest of George Dorr, who many of you are very familiar with, largely known as the father of Acadia National Park, that we relocated here to Mount Desert Island in 1921 at his request. He very much wanted to see a research laboratory here on Mount Desert Island where scientists from all over the world would come to study nature. And we have very much stayed true to that original vision of Dorr. Today we do a tremendous amount of research in a variety of areas, but our primary focus is on regeneration and aging. And Dr. Haller is going to tell you a little bit more about that and a little bit more about our unique approach, which is to study a variety of organisms in nature in order to understand more about our own biology and how we can maintain our health. So I do just mention that today in the Catherine Davis Center for Regenerative Medicine and Biology, we have a robust faculty of 10 full-time research groups. Our staff size is now around 72 employees. So we've grown a tremendous amount over the period of time that I've been here. And largely that's due to the tremendous support that we've received from many of you. So our sincere thanks to you for that. In addition to the tremendous research that's happening here, we are also a place of hands-on learning and scientific education. We have about 700 students a year that go through our various educational programs, and that is just a core part of who we are and what we do, that idea of shared learning, of bringing experts from around the world here to train the next generation of scientists, of physicians, and entrepreneurs. So that is a big part of what we do. In 2018, we were very excited to announce the appointment of Dr. Hermann Haller as our next president. And for those of you who have not yet had a chance to meet him, you will do so today, but we also hope you'll do that in person as soon as we're able. As we've discussed, he is highly esteemed for his work as a physician scientist. He's a nephrologist specializing in kidney disease and diabetic complications. He's a full professor and chairman of the Division of Nephrology at Hannover Medical School in Hannover, Germany. And he brings with him an extensive knowledge and appreciation for the work of the MDI Biological Laboratory. He's been a visiting scientist here since 1998, and he's maintained a full research laboratory here since 2007. So he knows us very well. He's very dedicated to the work of this organization. Dr. Haller received his medical degree from the Free University of Berlin, and he completed his postdoctoral work at Yale University. He's published more than 700 papers. He holds 6 worldwide patents, and he has founded 4 biotechnology companies. He's received many honors and awards and serves on numerous advisory boards, including those of Bayer, Boehringer Ingelheim, Genzyme, and Novo Nordisk. So it's my pleasure to introduce you to him today, to share not only his enthusiasm for the work here at the MDI Biological Lab, but his personal experience in treating patients suffering from complications from COVID-19 and in conducting research to understand the biology of this disease and to help us develop potential treatments that will minimize the threat of this devastating pandemic. So Dr. Haller, welcome.
Thank you, Terry, for this wonderful introduction. I hope you can hear me all, and I hope you can see me. I'm going to share my screen in a moment, and then we'll start the lecture. Let's see. What you can see here on the opening screen is a structure I'll talk about later on. What you see here is a cell surface in light green, and in dark blue, you see proteins sticking out from the cell surface. Now, this is our normal understanding of what cell surfaces look like. We have a membrane, which is mostly lipids, and then we have proteins, and they are swimming in the lipid bilayer of the cell membrane. However, what you see here also are these structures, these big structures in light blue sitting on and around the proteins, and these are sugars. And most people don't know that really, that proteins are covered in sugars. And what distinguishes proteins is not only their gene sequence, but the way they are covered in sugars. What you see also in red are very specific binding sites for other proteins on these sugar surfaces. So what makes us distinct from each other is on the one hand the proteins, but then very much the sugar structures we have in our body. And this will be a topic I'll talk about later on. As Jerry already mentioned, As Jerry already mentioned, I'm a clinician scientist. I've been coming to Mount Desert Island for 28 years. Actually, I came in the 80s, and I met John Forrest in 88, I think, for the first time. But I've been coming back regularly for more than 20 years. And Jerry, I think we both joined the lab, so to speak, the same year. And while Jerry has not changed at all, I have gotten more white hairs and I look different. And Jerry also mentioned that at the moment I'm president of MDIBL. I run a full-time laboratory at the lab, and we're doing vascular research, and I'm interested in kidney research as well. But I'm also a medical doctor. I'm chief of the department of the division of nephrology at Hannover Medical School, and I'm seeing regularly patients. I'm doing rounds, and it has been a great pleasure to combine these two sides of my profession. On the one hand, diagnosing and treating patients, being interested in patients, and on the other hand, understanding the mechanisms of disease. And going between these two sides of the same coin, basically, has been what I've been doing for the last 45 years. I've seen a lot of patients with COVID-19. I've seen patients on ICU, but I've seen patients with other lighter forms of the disease, and we have started to do research there, and all of a sudden it turns out that research I've been doing over the last couple of years is in a way associated with the mechanisms we're dealing with at the moment in our patients. And I'll use this example to talk both about my experience with patients and the research we're doing at MDIBL. We have published last week from Hannover Medical School our observations in patients with COVID-19. And what we did is we found, looking into the lungs of the patient, but also in other organs, that, and we are not the only ones, that COVID-19 is a disease not of the lungs. It's not so much the bronchial system which is affected, but it's the vasculature. It's a vascular disease. The virus enters the body through the airways, yes, but then it goes into the vasculature, it's transported into other organs, the kidneys, back to the lungs, but also to the gut. And the severe forms of the disease happens in patients when the other organs are affected. Very early on in the disease, we have seen that kidneys are affected, and there is a large study from New York City where they looked and they found that very early on you can detect changes in the functioning of the kidney in patients with the severe form of COVID-19. Now, when we look into this, and these are pictures from the autopsy of a patient from a lung who died from COVID-19, you can see immediately that there is a lot of inflammation, especially on the lower slide. You can see lots of infiltrates. There are inflammatory cells in there. And when we go a little bit deeper and we look into the blood vessels in the lung of this patient, what you see here, you can see thrombi, so clots of blood platelets, and you can see erythrocytes. These are here in red. So we have a disturbance of blood flow. We have microthrombi, we have clotting inside the blood vessels, and this is what makes the disease so serious. So most of you have heard that patients have been, when they have the serious form of the disease on a ventilator for days and weeks, and this is because the blood vessels are clotted, not only in the lung, but also in other organs. And the clotting of blood vessels, this is mediated by endothelial cells. So endothelial cells are lining, you can see this here, the blood vessels. And all of a sudden, although I'm not a virologist, and to be honest, my interest in infectious disease has been limited, all of a sudden COVID-19 is a microvascular disease, and this has been my arena, and we have been doing a lot of research in the microcirculation over the last 20 years. So we have a disease which starts with bronchial symptoms. We have an infectious disease where we take up a virus which then enters the bloodstream and leads to microvascular disease. It's not that astonishing. Other viral diseases are doing the same thing. The most severe disease, viral disease we know is Ebola virus, and there you have massive rupture of small blood vessels, you have bleeding and a lot of leakage. COVID-19 is not as severe, but you have a massive infiltration of the endothelial cells. And here you see from our patient electron microscopy pictures of these endothelial cells which line the blood vessels. And you may appreciate that these endothelial cells, which are normally very flat, they are turbulent, they are activated, they really try to form new blood vessels. They are desperately trying to overcome the damage which happens through the viruses which have entered the endothelial cell. So the activation of the endothelial cells, this has been one of our research topics, and I'll get back to this in a moment. Now, we have found the virus not only in the kidneys and in the lungs, but we have found it in the gut. And Klaus Stahl, who was a student of us at MDIBL, he has been working with John Forrest for 3 long summers. His brother is, his twin brother is chief resident at Yale. And Klaus published this letter in the Lancet also last week, where he found that SARS virus is in the gut. This was also a patient who was severely sick, who actually had lung disease for 2 weeks, excuse me, got better, came back 3 weeks later with severe disease, microcirculation disturbances in the gut. And Klaus could actually find the virus in the endothelial cells of the gut and published this in the Lancet. You can see on the left-hand side a quotation from Klaus. He was a wonderful student. We still remember him vividly and his activity. And he said, the 3 summers I spent at the MDI Biological Laboratory was the beginning of everything for me. He's now more clinical. He's not that much in the lab, but his curiosity, and he's still a scientist. He publishes, and you can see this example from the patient with COVID-19 as a very nice example. Now let's go back to the endothelial cell surface and the way the virus enters the endothelial cells. You can see here on a complicated slide, two receptors. One is the so-called ACE2 receptor, which plays a role in vascular biology. And this is the receptor which has been discussed a lot. It binds the virus and it internalizes the virus. So it's an essential, important receptor for bringing the virus into the endothelial cells. However, before ACE2 binds the virus associates with these sugar structures. You can see here what I've shown you on the first slide, a protein which is anchored in the cell membrane, and you have heparan sulfates. These are the sugar chains you have seen in a different form before. And the virus associates first very specifically with these sugar structures, very specifically with certain heparan sulfates. And when you cleave these heparan sulfates, and you don't have them anymore on the surface, the uptake of the virus goes down by 80%. So we are understanding how the virus infects cells, and this brings us then to a better understanding how to develop strategies to prevent that. For us, it has been fascinating because although I said this before, I have not been a virologist, all of a sudden a structure which has been very dear to my researcher's heart over the last 20 years, and these are the sugar structures, they play an important role in the uptake of the virus. You can see this in the next slide. This is from our research at MDIBL. Sorry. There was a question? No. You can see here these proteins, the sugar structures. A lot of molecules are binding to these heparan sulfates, and we have been investigating this, and we have inhibitors to prevent this binding. When I go back to the last slide, we have inhibitors, quite a few of them, which inhibits molecules binding to heparan sulfates, and now we are testing, obviously, the hypothesis that some of these small molecules may inhibit the virus. And we are carrying out very interesting experiments at the moment to identify small substances which can be swallowed. So they are oral medications, and they already have been used in humans. So it may well be that within a couple of months, we are able to prevent the uptake of virus particles on the endothelial cell surface. We can visualize that. And this is now Mount Desert Island Biological Laboratory. What you see here in green are endothelial cells, and what you see in red is actually the sugar structure on living endothelial cells using electron microscopy. And this is one of the areas we want to be world-renowned at MDIBL, visualizing what's going on in biological systems, trying to understand molecular mechanisms and cellular mechanisms of biology and of disease by bringing this into a form which can be seen by the public. Because we think that in order to understand science, you have to see it, and this helps the understanding. So are there any questions? Because we will now go into MDIBL, as you can see here, about the mechanisms we have just been discussed. Are there questions about COVID-19? We can answer this at the end of the lecture, or we can do this in between.
Oh, can I, Kathy Vino, I'm a neophyte at this. Could you explain a little more about what you mean by the sugars in the body and heparin sulfites, and what sugars are these?
Yeah, that's a very good question. I try to be hopefully very clear and short. It's not the sugars which we eat. It's when proteins are made in the cells add sugars to the surface of these proteins in order to distinguish one protein from the other. So for instance, where all the time where we have lots of different proteins, which only small changes, like antibodies, for instance, the cells add sugars to these proteins, and these are the sugars, the heparin sulfates I'm talking about. We have a variety of these sugars. When they're on the surface, they're called glycocalyx. So it's a biological property of a lot of proteins that they have sugar added to the protein to distinguish it. And these sugars can be used by viruses to bind to the surface of cells, and then they are handed over to the next receptor, and then they're taken up. So it's not it's an enzymatic, that's what we called it, an enzymatic glycosylation. So it's a normal behavior of cells. When you have diabetes, you have too much blood sugar, and the sugar also binds to proteins, but in a disorganized way, and we call this non-enzymatic glycosylation.
So I have a question. My name's Randy. I've read recently that losartan, a blood pressure medication, may actually compete for binding at the ACE2 inhibitor, or at the ACE2, I mean. Do you know anything about that?
Yes, that's true. So in the beginning, there was a lot of speculation because there is interaction with the ACE2 and sartans, for instance, like losartan, whether they may be not beneficial and may even help the uptake of the virus. These were early speculations, and then all international societies came up with a recommendation: do not stop taking your sartans. And from what I've been explaining, the important receptor is heparin sulfates, and ACE2 is only a second receptor. However, it may well be that we will find also inhibitory molecules. Our neighbors on MDI, the Jackson Laboratory, they are just generating a mouse without the ACE2 receptor as a model how to treat the disease.
Great, thank you.
Dr. Haller, we have a couple of questions in the chat. One here is there any variance in the patient's DNA that explains the severity of their disease, and do the genes have anything to do with their cell surface molecules?
At the moment, we have no evidence that the genetic profile of the patients make them more susceptible. It may well be, from what I've explained, that there will be in the future molecular changes. At the moment, the best explanation we have to explain the severity of the disease, when you have viruses in your microcirculation and you already have damaged blood vessels, then you're more prone to severe disease. You know, it makes a lot of sense. You have the virus, if you have a young endothelium, if you have a young microvasculature, they can take up the virus and they don't respond that seriously to the virus. When you have already damaged endothelium, and this is when you are older, when you have hypertension, when you have diabetes, you may have more damage to your vasculature, and then you get the infection, and the combination of that may lead to more serious disease.
Fascinating.
They-
And one more question in the chat. Are there differences in heparin sulfates between genders or across age span that could explain differences between COVID-19 morbidity rates,
Yes.
especially between men and women based on age? Or are they expressed differently in people with high-risk conditions that would render more susceptible? So you've addressed some of that. But.
Wonderful question. We could recruit you as a postdoc at MDIBL because this is so far a totally unsolved question, whether the heparin sulfate pattern, we know that it changes with age, but how it changes and which enzymes are responsible, that is virtually unknown. And we have a new animal model where we want to test this hypothesis over the next 2 years. The sugar coating, is it different when you age? In addition, we have some evidence that it's different in women, but so far only in autoimmunity. So the antibodies may be different with regard to the sugar structure. We don't know anything at the moment about the sugar coating of endothelial cells in men and women and whether it's different.
And just one final, you've addressed some of this, but says, Dr. Haller, can you use this insight to conjecture on why COVID-19 is so selective, causing severe illness in some and trivial symptoms in others?
Yes. As always, it's a cascade of events, but it explains why elderly people and people with diabetes and hypertension are more affected, because they already have damaged blood vessels because of the underlying disease. It's not happening in everybody, but I think it's the best explanation at the moment to explain the damage to the microvasculature. If this is already present when you're exposed to the virus, especially a heavy virus load, then you have more severe disease.
May I ask a question, Hermann?
Yeah.
Which has to do with the cytokine storm and the relation between the cytokine storm in adults and children who seem to have very little symptoms at first until it overwhelms them. So I'm just, can you explain that?
Yes. When you have seen these endothelial cells in the electron microscopy pictures I've shown you, that they are really very active. They are becoming very activated by the virus. So they are functionally very active, and they produce all types of cytokines and inflammatory cytokines. And when you already have diabetes, you already have a low level of cytokines. When you have hypertension, you have low levels of cytokines. And as I said before, you add the virus, and then it's an overwhelming cascade of the release of cytokine. We can measure this in patients. We can measure interleukin-6 and other cytokines, and they are elevated in all patients with more severe forms of the disease.
And children?
And children, their endothelium is more innocent. You know, they have not seen that many damaging things happening to the endothelium. They haven't smoked so far. They have not a high blood pressure. Their endothelium is in much better shape than ours. And you can see this, you know, they heal better. They can't regenerate, unfortunately, but when they have a wound, it heals much better. The sprouting of the blood vessels, that's more active. It's better regulated than in older patients.
Just a couple more questions, Dr. Haller, that have come in. Sorry. One is, there's some new research, not yet peer-reviewed, that shows blood type may affect the course of the disease. Blood type A is the one that the research pointed out. Have you heard anything about this research?
Yes. At the moment, we are flooded with reports like that, and they are difficult to explain. If I use the explanations I've provided you with so far, blood types, whether you're A or B, this is also because of the sugar structure of the blood types. So it may well be that it has something to do with the glycosylation and the glycosylation pattern. But that's at the moment pure speculation on the basis what I've explained so far.
And so a similar question, a report that smokers actually may seem less susceptible. Is that sort of a similar situation?
Yes. Smokers when, now it gets complicated, but also interesting. You know, smoking is not all that bad for a specific purpose. It damages the surface of your bronchoalveolar cells in such a way that the glycocalyx, these sugar structures, are gone, and then you are not taking up the virus as good as when you don't smoke.
Oh, God.
However, when you smoke, you also damage your glycocalyx in your microvasculature, and then you have vascular disease. It's ironic, but when you have a lung transplant, you're better off when you smoke a little bit. It's not a recommendation, but it has the same effect on the surface of your alveolar cells. So there are some small positive aspects. And please don't tell anybody that I have said anything positive about smoking. My students will kill me.
Just let that not be the takeaway from this afternoon. A couple of other follow-up questions, and then I think we can get on with the rest of your presentation. But one is use of blood thinners in preventing ineffective treatment for clotting. Is there?
Yes, that's a very interesting question. You know, we have published our paper a week ago, so we have only now a couple of weeks of research into the microthrombi. We know that there is a disbalance of coagulation factors in these patients. So that's a very interesting question, whether we can use anticoagulants in order to prevent the disease. A little bit complicated, I mentioned our small molecules, which we use to prevent the binder to heparan sulfate. They also influence coagulation. So the heparan sulfates, these long sugar chains, also play an important role in the coagulation cascade. So it may well be that we can solve some of the problems with the microthrombi in the vascular tree by anticoagulants. But we'll see how this works in the future.
Great. Thank you. We have a few more questions that we'll hold till the end, maybe if you want to just continue with your presentation.
Okay. So you have seen this wonderful picture. This is what we want to do and what we want to achieve at MD-IBL. Here you see lovely MD-IBL, and most of you know the place. Not a lot of you have seen MD-IBL from this perspective. I think you have either to sit in a helicopter or you have a drone and look at the picture. But you see here our buildings. We are more than 100 years old, and we want to be the best. We are very focused. We want to bring research, very focused research, to MD-IBL, and we have been doing this over the last 100 years. We want, at the same time, to have a discussion. Science is about asking questions, and MD-IBL has been a special place where you work hard, but you also have time to ask questions. You communicate. You can stroll down to the sea and sit there, and it's not a bad thing for a researcher to think for half an hour overlooking Frenchman's Bay and then have a discussion with your colleagues on campus. And that's one of the important ideas of MD-IBL as well. And thirdly, we are teaching. We have a very long history, and you have seen this one example of Klaus Stahl, who had transformative summers at MDIBL. We have become a year-round institution, but we are keeping this tradition of curiosity, of communication, and of teaching. And now some of these young students are not spending their summers at MDIBL, but their winters. And it will be very interesting to see whether they are different when they spend a long winter at MDIBL and doing research there as compared to their summer colleagues. 120 years of discovery. And I believe that discoveries is what we want to have at MDIBL. You can see here Jessica Lewis on the left-hand side, and she was one of the first pioneers using cell culture in the US, importing it from Germany at the time and using it at MDIBL. And you see here over the last 120 years, lots of people meeting there. Here you can see me and a group of scientists 2 years ago. And these are our new research buildings. So this has all been built. We started 20 years ago, 22 years ago with all these buildings with the support of the Morris family and the Davis family. And these are the labs and these are the people we are carrying out research with. Here on the right-hand side, this is one of my favorite pictures, not only because I am on that picture, you can see me here right next to the whiteboard, but this is how we teach students. Not all the time. It's raining sometimes, and sometimes we are in the labs, but we are a meeting place, we are a research place, and we are a teaching place. What are we doing? What is the topic? We are interested in aging and regeneration. This shows you a painting by a German painter, Renaissance painting. And what you can see here is that old people are coming from the left-hand side and they are in the fountain of youth, and then they become much younger and they enjoy life again. And we are trying to understand what's happening in here. So we are trying to understand aging and we are trying to understand regeneration. It's exactly this blue line where we want to understand which factors, sorry about that, which factors affect aging and regeneration. So we are interested in environment, not only in the molecular mechanisms, but how environment affects the mechanisms of aging and regeneration. And then last but not least, we are interested in biomedicine and we are interested in therapies. We want to use what we find and what we understand and use these as therapeutic strategies. We understand that's a long way from understanding of molecular mechanisms into therapy. You need much bigger institutions for that, but MDIBL is the place where the spark starts and where we start thinking about the mechanisms and the therapy. We use animal models, and we have a long history in that. Since the times of Darwin, it has become clear that in order to understand mechanisms of disease in humans and mechanisms of physiology in humans, you have to go to the ocean where you find lots of animals, and this is why we are a marine biology lab. But these days we have more animals, and we are not only taking them from the ocean, but we have fish, zebrafish here. We have killifish here. These are African killifish, a model of aging. We have C. elegans, a worm, which is a model for aging as well. We have Drosophila, and we have axolotls. You don't want to know how many axolotls we have. At the moment, we have more than 900, and in half a year we'll have 1,700 axolotls, because we are very interested in limb regeneration in these animals. And we want to compare the mechanisms in these different animals. We are a unique place. There are not that many academic institutions where people are working shoulder to shoulder with different animal models and ask the same questions. And we need technology for that. And very important is bioinformatics and artificial intelligence. There's a strong movement worldwide to use artificial intelligence to understand all the data. And you can see here examples. We are not looking at tissue anymore. We are dissecting tissue into single cells, and then we analyze the gene expression in these single cells, and this produces tons of data. And we have two aims. We want to develop novel methodology to understand this and to use this, and we also want to teach. Worldwide, there is a shortage of young people being trained in bioinformatics. In Portland, in Maine, next year, there will be a new institute with a lot of money supporting the education of students in artificial intelligence. And we and the Jackson Lab and the College of the Atlantic want to be part of that. And the second technological development at MDIBL is microscopy. We are installing, even at COVID-19 times, a new microscope, 1.2 million. It's expensive. Science these days is not easy to have. And we can use this microscope to generate these pictures you have seen in my lecture before. So bioinformatics on the one hand, and microscopy. So seeing is understanding, and using artificial intelligence in order to create something useful out of the chaos of such a lot of numbers we are producing. That's our other goal. We want to regenerate organs. That's one of my favorite pictures, because these days, not all of our students are so enthusiastic. The US has flown to the moon 60 years ago, and nobody actually believed it in the first place. And when you compare 747 with Kitty Hawk, you can see this is only 120 years. This is how we think we can contribute to the development of organs and regeneration of organs. And what you see here, in addition to that, is here the structure of a kidney. Almost unbelievable that we will be able to generate this, but we have organoids, we have stem cells at MDIBL, and we are trying to understand the mechanisms using the tools you have seen before, and then we use these approaches in order to make new organs. We have a wonderful faculty. We have started more than 12 years ago to be year-round, and over the last 2 to 3 years, we have recruited Ian Drummond from Harvard, one of the world-renowned specialists in zebrafish. We are recruiting at the moment Briak Muravala from Vienna with all the axolotls, and you can see that we already have other researchers here, and Briak, together with James Godwin, they will form a team where all of a sudden MDIBL is on the forefront of limb regeneration. Ian and I are working together on kidney in zebrafish. Eric and Jared and Sam Beck, they are experts in aging, mechanisms of aging, protein translation and aging. So we try to recruit the best, but we also try to form teams to be competitive in the future. we are all together in this wonderful campus, but we also use our campus to invite the world to MDIBL. MDIBL has, and I'm a very good example of that, a long tradition in international exchange. As I said, I came to MDIBL first time in 1988, and then I came back in 1998, and I stayed. And I'm stepping down at Hannover Medical School, and I'll be full-time president shortly at MDIBL, but we'll keep these international connections. We want the world to come to MDIBL. And to be honest, it was just mentioned that we are a well-kept secret. I can tell you, we are not a well-kept secret in the international world of science. In the kidney world, for instance, MDIBL is holy grounds. We have people coming to MDIBL and they understand what the contributions of MDIBL has been and hopefully will be in the future. We need your support for that. We have talked about technology. We have talked about recruitment of new faculty. And it's not that easy, and it needs a lot of support to move somebody who is world famous from Boston to MDI. You can see that natural beauty is part of that, but they come for research. And it's not easy to bring people from other parts of the world, from Austria, from India to MDIBL. So we are very thankful and grateful for your support, because without your support, we would not be able to do all this research and to contribute. Thank you very much.
Thank you, Dr. Haller, so much. Thank you for sharing your insights about this complicated disease and helping us to understand it a little bit better. There certainly seems to be a lot of good that's coming out of this understanding, and hopefully soon will directly contribute to eradicating this disease. So thank you so much. There may be a few more questions here, I think, before we wrap up. And let me just see, we have one question. Some patients with COVID-19 who recuperate when tested afterwards test negative and then positive to COVID back and forth. How long on average does the body take to completely eradicate COVID-19 and to heal the damage in the vascular system?
Yeah, these are very good questions. And to be honest, we do not know a lot about that. You know that testing has not been available to everybody, and testing has been done in some patients, and we have found that sometimes spouses, one is positive, and the spouse is negative and is testing negative for the next couple of days. We know that sometimes we test negative. The question is always, is this because we have not done the swabs correctly? You know, because the virus is not everywhere. We have to go up into the nose, and we have to do deep swabs in order to get the virus. So there may be a variability. We know that the virus can come back. These are rare cases. Don't be afraid of that. When you're exposed for the first time, you are immune afterwards. However, we do not know so far how long the immunity will last. So for a medical doctor, there are still a lot of questions. We are having a new disease. We are learning from the disease. For us, it will not be over. It will be a disease in the textbooks. We'll see different presentations by patients. However, what will be over is the pandemic. I expect in the future, with all the precautions we have at the moment, that we will have infections now and then. It may be even in larger groups, but the pandemic will be over both in Europe, where we can see this already, and in the US as well.
Thank you. And of course, the big question, any comments on the timeline for a vaccine?
Well, the good thing is that there were already some efforts underway. Bill Gates was supporting this, and they were working on coronavirus vaccines. So we had a head start. There are now a lot of RNA technologies around, so this makes it much easier to generate a vaccine. So these are the positive points. And at the same time, a lot of energy went into that. There are now really dozens of companies around the globe working on that. The big question is the testing of the vaccine. I mean, you have to test in patients to see whether it's working. And these clinical tests are at the moment underway. Me personally, I think we will have a vaccine before the end of the year. However, it'll take another 3 months or so to produce the amounts necessary that it can be used in patients on a large scale. So I think by next spring, we have to survive the winter, I think, without a vaccine.
Mm-hmm. And Dr. Haller, Europe is a bit ahead of us here in the US in terms of reopening. Tell us your thoughts about the possibility of a second wave, or what you think of that potential.
These are the conclusions, and we already can draw these conclusions from the experience we have worldwide and the experience we have in Europe. The outbreak, the pandemic happened because people came together in these early days of the pandemic without any precautions. We had, for instance, in Italy, northern Italy, some of you may remember Bergamo was the city where a lot of COVID-19 happened. They had a Champions League soccer game during the first week when they had infection all over Bergamo. 90,000 people, Italians, met for one evening in the local stadium because they were playing Barcelona or Madrid, I don't remember. Hugging, drinking, having a great time together, and then a lot of infection. And in the big cities, you know this, in New York City, you can't escape. I mean, in the underground, so when people come together in large crowds, this is when you get infected. When you have distance, now you wear a mask, it's highly unlikely that there will be a second wave. You know, this is over, and we can see this in Germany. The numbers are really going down. Life is coming back to normal. However, distancing, and when you are in public spaces together with other people wearing a mask, this prevents most of the infections. There is still a residual risk, but for the time being, it's distancing, wearing a mask, and disinfect your hands. And these are the rules we have established at MD-IBL. We have been following these rules, and although we are a little bit afraid of all the tourists coming to Maine now, I think it'll work, and it'll prevent the spreading of the disease almost completely.
That's good news. Now we all need to just behave ourselves. That's great.
Yeah, I can tell you, I've been swimming with my wife yesterday, public swimming pool, and everybody's swimming without a mask, and people are keeping their distance. And I start swimming, and an elderly lady stops and says, I don't want to be your teacher, but you're swimming this way on the right-hand side, and when you come back, please use the left lane. So you have to change your behavior, but otherwise life is almost normal.
That's great. Well, Dr. Haller, thank you so much for sharing your perspectives with us today. We really appreciate it. I think I see a lot of positive comments in the chat, thanking you for your time and for the insights. And so Really want to extend our appreciation to you and to all of you for joining us today. And it was wonderful to see you, but also wonderful to have an opportunity to share some of the exciting research that's happening here at the MDI Biological Lab, as well as our vision for the future and what we hope we will be able to accomplish with your help going forward. So for those of you who are just getting to know about us, we encourage you to continue and join us in the conversation as we talk about different aspects of the laboratory's work over the next few months. We hope to have more of these kinds of gatherings, and maybe eventually we'll be able to get together in person, and we really look forward to sharing those times with you. We'll be sharing a lot of other events that we have planned for the summer. All of the information for that is on our website at mdibl.org/events. For those of you who are members of the Star Point Society, there will be other exclusive opportunities, and we look forward to sharing those with you. So again, if you're interested in learning more about the Star Point Society and the benefits of membership, please check out our website. So thank you all. Jane, or Anna, or Rob, any final comments wrapping up?
No, I was just saying thank you all, and thank you, Dr. Haller.
Wonderful.
Thank you.
All right. Thanks, everyone.